Good evening. My name is James Garner. I'm the Chief Executive Officer of Antisense Therapeutics, and I'm really grateful to be able to welcome you all to our inaugural shareholder open house meeting. This is the first time we're doing this, and so I wanted to maybe just take a minute or two to explain the format a little bit. One of the things you'll notice over the next few months is we're really going to be trying to make an effort to reach out to all our shareholders in a whole variety of different ways. But this is a really important part of that, because I think there is no substitute for being able to meet our shareholders face to face and being able to just discuss the progress of the company with you live. So we're doing one here in Melbourne. We'll be doing one in Sydney next week, and hopefully in the future we can take this out to other parts of the country. And in the future we might zoom in on particular aspects of the business. But today, since, as you know, I'm pretty new to the organization, and I think we've got a few new shareholders as well, I thought that we would just really take it from first principles and go through the company's story fresh. And I think for some people in the room, there'll be some familiar points in here, but hopefully there'll be some points of interest for everybody. So I'm going to speak for about 20-25 minutes, and I really love to address as many questions as possible from everybody. So please ask away when we get to the time. So let's get started. I put up here to start with a kind of executive summary of where the company starts. And to me, this crystallizes the investment rationale for Antisense in one slide. And as I've been going around meeting with investors over the past few weeks, and as I continue to do so in the weeks ahead, there's really almost just 3 points here that I want to get into people's head. 3 key takeaways, if you like. And they're these. First of all, the company has a really strong asset in ATL-1102. This is a drug that has a great deal of scientific potential and a great deal of promise. It's a well-proven technology, antisense oligonucleotides, which we'll talk about in a minute. We've seen some really promising data out of the earlier phase 2A study, and we are now in a really, really well-designed study that should give this drug every chance to prove its credentials. So we've got a tremendously strong starting point there. If we are successful, the prize ahead of us is very substantial. The second point here, the commercial opportunity for ATL-1102 is large. Duchenne muscular dystrophy is what is often described as a rare disease, and it is in comparison to hypertension or type 2 diabetes. But that really belies the fact that there is an enormous commercial opportunity here. The pricing for drugs like this, the economics of bringing a drug like this to the market are very, very attractive for a company of our size. And then finally, I think the company is now extremely well placed to deliver on this potential. We have a terrific team, of which we'll talk about more in a few moments. And thanks to our recent financing activity, we're well funded to do this work. As I think everybody in the room is aware, we recently raised $8.35 million in gross proceeds from a placement to sophisticated and institutional investors. And we followed that with a share purchase plan that raised $3.26 million from shareholders. So we're in the enviable position for a biotech company of having money in the bank to do the work we need to do. We can focus on the drug development instead of worrying quite so much about how we're going to pay for it. So that, in a way, is really the message I want to get across this evening, but let's go into a little bit more detail. And I want to start really zoomed out here and touch on the pipeline of the company as it stands today. And we'll mainly focus on Duchenne muscular dystrophy today because, of course, that is the focus of the company. But I did want to reflect that really one of the things that attracted me into the company when I joined a few weeks ago is actually the breadth of opportunity that ATL-1102 represents. Although our focus is Duchenne's disease, we're in a phase 2B clinical trial, as everybody knows, which we'll talk more in a few minutes. We have other work going on. We're already looking at other aspects of Duchenne's treatment in combination with other kinds of therapy in earlier stage patients. We have some really interesting work going on in a disease called limb-girdle muscular dystrophy, R2, also known as dysferlinopathy, and we should hopefully be able to share some information about that pretty soon. And then historically, the company's looked at multiple sclerosis with this drug, and indeed there's some other indications we've been tinkering with behind the scenes. ATL1102 is really what is often described in the industry as a pipeline in a molecule. It's a drug that has enormous potential across a whole range of different therapeutic areas. And then, of course, we have a second asset in ATL1103. Now, we've got some thinking to do among ourselves over the next few months, how we balance our focus, how we prioritize all these opportunities. And for right now, as the chair of our board, Dr. Charmaine Gittleson, is fond of saying, we are laser focused on Duchenne muscular dystrophy. But I think it's important just to start by reflecting that actually we have an embarrassment of riches in our current pipeline. Let's talk, though, about Duchenne's disease. And again, apologize for those in the room for who this is familiar, but look, as an introduction to Duchenne's, well, this is about as tough a disease as you'll ever come across. This is a genetic disease that affects kids. It's on the X chromosome, which means it is almost exclusively a disease of boys rather than girls. And fundamentally what happens in patients with Duchenne's disease is they are unable to make a protein called dystrophin. Now, dystrophin is found in your muscles, and it essentially stops the muscles from damaging themselves as they relax and contract. So if you can't make dystrophin, your muscles basically damage themselves every time you move, and over time that damage builds up and leads to a lot of fibrosis and scarring and fatty infiltration, and these kids gradually lose muscle strength. It's a little bit like in a car, if you don't put oil in the engine, all the cogs and the pistons and the cranks and things all kind of seize up and destroy each other. It's a little bit like that in the muscles of children with DMD. And so unfortunately, these kids by their teens are usually wheelchair bound, and unfortunately life expectancy at the moment is typically in the early to mid-20s. So this is a tough disease space to be working in, as you can imagine. How do we treat this right now? Well, there's two fundamental ways that we can treat Duchenne's disease. Now, obviously, if the problem is that you're not making dystrophin, one approach is to try and restore dystrophin, to try and help these patients make dystrophin. And this has been a very active area of research in the last few years. We've seen several companies bring forward what we might call for today dystrophin restoration therapies. And you can see some of the main ones up there in the top right of the slide. Two problems with dystrophin restoration therapies. One is that they're very sensitive to the individual genetic mutations that these kids have. Duchenne's disease is not one disease. It's actually a complex of hundreds of different genetic abnormalities underlying it. And these drugs tend to be very, very specific for individual genetic profiles. So most of them are only really applicable to somewhere between about 8 and 12% of patients. And as of today, the majority of Duchenne's patients do not have a dystrophin restoration therapy or exon skipping therapy indicated for them. And the second problem with these drugs is that although it makes enormous sense, not enough dystrophin, so let's fix that and make more dystrophin. In practice, these drugs are not wonderfully efficacious for most patients. The clinical efficacy has not been stellar. And for those that follow the detail of these things, some of the discussions with regulatory agencies like FDA have been a little contentious. So this is by no means the last word in the treatment of DMD. So the other plank to treatment here, as you can see on the lower part of the slide, is that we've looked at ways to reduce the inflammation that is associated with this disease. We've looked at various kinds of anti-inflammatory drugs. Now, indeed, the mainstay of treatment has been a class of drugs called steroids. These are anti-inflammatory drugs we use in a whole range of different illnesses. And we've had some promising results with steroids, but they're a class of drugs you don't want to take for a long period of time. There's some quite considerable toxicity here. So in recent years, we've seen other kinds of anti-inflammatory drugs coming along, but this is really where ATL1102 sits. This is a drug that acts by controlling the immune system, by reducing inflammation, and because of that, it's a drug that is potentially applicable for almost all patients with Duchenne disease. The other great advantage here is that it is a highly combinable drug with other kinds of therapies. So when we look at a slide like this, we don't really see competitors on our slide. We see drugs that hopefully in the fullness of time, our drug can be co-administered with. And we'll talk about that a little bit more in a moment. That's where we stand now. How does it work? Well, look, there's an enormous amount of very technical science here, which I'm not going to go into in great detail, but let me just say for today that ATL1102 is part of a class of medicines called antisense oligonucleotides. And what does that mean? Well, in our bodies, most of the structure and function of our body is driven by proteins. So, for example, the collagen in our skin is a protein. Insulin, which controls our blood sugar, is a protein. The instructions to make proteins are coded in our DNA. And in between the DNA and the proteins, there's a sort of translational molecule called RNA. And so one of the things we all learn at first year of medical school is DNA makes RNA makes protein. Now, antisense oligonucleotides interfere with this process in various ways. Now, sometimes they interfere in such a way as to correct the manufacture of proteins. And so, in fact, some of the drugs we mentioned before, some of these so-called exon-skipping therapies in Duchenne's disease, they actually work to try and correct the dystrophin that is produced. They try and make proper dystrophin. Our drug works very differently. We're actually not targeting dystrophin at all. We're targeting a protein called CD49D. It's one of those wonderful names that only a molecular biologist could come up with. CD49D is part of our immune system, and it controls the way that white blood cells move into tissues. So it's a very, very important part of inflammatory responses. If we block CD49D, which is what 1102 does, we reduce white blood cells moving into tissues, and we reduce the inflammation that they cause when they get there. So fundamentally, this is a broadly active anti-inflammatory drug, and it hits right at the point where Duchenne's is operating. I mentioned that it works as an antisense oligonucleotide. I just want to say in passing, it is not the first drug to do this. This is a very well-proven technology platform. Depending a little bit on how you count it and where you draw the boundary, there are 9 FDA-approved antisense oligonucleotide therapies, and they're shown down the bottom of this slide. So this is a pretty well-proven technology space. Clinicians, regulators, investors are used to seeing these kind of drugs. We're the only one in this space targeting CD49D, but we're not reinventing the wheel here. We're using a very well-proven technology space. So that's the theory. How does this work in practice? Well, this is the data from our phase 2A study, which was completed a couple of years ago. And I must admit, when I saw all this data in one place, it really put the hairs up on the back of my neck, because I think this is actually fantastic data for a drug at an early stage of development. The first thing I want to draw everybody's attention to is the top line here, which is a measure called PUL 2.0. It stands for power in the upper limb. It's a measure of, as you can imagine, how well the patient's arm works. And it looks at the shoulder, the lower arm, and the hand. This is a key measure in Duchenne's disease of how patients are doing. When we gave these patients ATL1102 for 6 months, we saw actually a little improvement in their PUL 2.0 score. It improved by 0.9 points. Now you might say, well, that sounds great, but what happens otherwise? What happens if they don't have the drug? And if you look at historical controls, if you look at benchmarks here, if you look at what happens to very, very similar patients who don't have the drug, the equivalent data is that they deteriorate by around 2 points. So in other words, there's about a 2.9 point difference here between the patients that were treated and what we would expect to happen in the absence of treatment. That's quite substantial in this game. And look, I'll be quite honest and say if we were able to replicate just that data point in our phase 2B study, I don't really have any question in my mind that we would have a very viable commercial product on our hands. So that alone is very promising data. But that's not all we have. You can see there a bunch of other functional measures, myogrip, myopinch, and Moviplate. These are other measures of function. As you can see in general, the patients treated with ATL1102 either improved a little bit or at least stayed stable. And in general, the patients in our comparison group didn't do so well. The Moviplate one's a little kind of quirky exception to the rule here, but honestly, it's nobody really knows quite what that means in DMD. So this all looks really good, but that's all a somewhat subjective measure of how patients are doing. We also looked at MRI scans, and that's not subjective at all. That's very, very objective. And again, we look at the lean muscle mass in the child's arm. That's a very, very good measure of how their disease is doing. A little bit of improvement in the 1102 group and deterioration in the comparison group. And finally, lymphocyte counts, white blood cell counts. That's a measure of whether the drug is acting as an anti-inflammatory drug. Again, they go down a little bit with ATL1102, which is what we would want to happen. And in the patients who don't get treatment, it goes up. So any one of these data points in isolation would be very promising. Taken in aggregate, they really triangulate a drug that I think is unquestionably active. So this is a very exciting set of data. And as I say, if this can be replicated, we are off to the races. Now, two caveats here, because I want to be very open with you all. This is still a smallish study. There were 9 patients here. And actually, that's not that tiny by the standards of a disease like this, but still doesn't give us quite as much as we need. So we need to replicate this in a larger study. And secondly, this was a single-arm study. All our patients got the drug. And so we have to compare against comparators outside of the study. And I think regulators are going to expect us to compare against patients within the study. So what we need to do now is replicate this kind of data in a larger randomized controlled trial. So that's exactly what we're doing. And this study started recruiting in June. We're recruiting patients almost exactly the same as our phase 2A study. So these are, in particular, non-ambulant kids. That is to say, for practical purposes, kids in a wheelchair. They're relatively advanced in the disease, but otherwise, you know, really, this is a very, very similar population to our phase 2A study. We randomized them to get either placebo or one of 2 doses of the drug. And after 6 months, we're going to look at their POOL 2.0 score. So this is a fast study. We're aiming to complete recruitment by the end of this year, and then we'll have a primary endpoint 6 months after that. So, so this is pretty brisk. Now, we'll then follow the patients for another 10 months to get some long-term data, which is something we think will be required for regulators. But the primary endpoint, 6 months after the last patient is in. Now, where do we go after that? Well, look, I won't go into a huge amount of detail here. I'm not going to labor the point about all the different regulatory pathways this drug could follow, but suffice to say, there are a lot of ways this drug could find its way to market. Very, very broadly, the question is going to be, is the phase 2B study itself sufficient to get some kind of marketing authorization in some form, or will regulatory agencies expect us to do a little bit more? And you know, we'll have that conversation when we have data in our hand. One really important nuance here, though, that I do want to stress here today, though, is that any scenario on this slide that has a green box, in other words, any scenario in which the drug reaches market, we have the ability, in principle, to achieve a pediatric priority review voucher. Now, what is this? Well, this is a kind of golden ticket that FDA offers as an incentive to companies to develop drugs for childhood diseases. And the holder of a PRV can get FDA to review any new drug in 6 months rather than the usual 12 to 14 months. Now you might say, well, what does that mean? Well, if you've got $1 billion drug, you know, a drug with $1 billion of expected sales, and you can shave 6 months off your FDA review, well, that's worth about $500 million in revenue. So these are really kind of valuable, these tickets. Now, we don't have to be the ones to use it. We can sell it, and any holder can use this voucher. And the going rate for these things is about $105 to $110 million. I actually know the investment banker in New York that does most of these deals, and he reckons he can sell one in about 6 weeks. So there is a real demand for these priority review vouchers. If we can get 1102 approved in any shape or form, we immediately have an incredibly valuable asset in our hands before we sell our first unit of product. So I think this is a really important nuance around the drug. But nevertheless, what does 1102 look like as a commercial product? Well, there's some early answers to that question here. And as you can see, this is by any standard of reckoning, an extremely attractive commercial market. So on the left here, you can see some of the comparator drugs that are out there. Many of these drugs are only approved in single territories, and they're either approved all in the US or just in Europe. So these are often kind of single territory figures for their net sales. But you can see this is rapidly growing, 34% year-on-year growth. And when you look at pricing in the middle column, what you see is pricing typical of therapies in these kind of rare disease indications. Very, very compelling price points for new therapy. And certainly our internal estimates is very, very preliminary work, and this is just putting a finger in the air, but we figure on ATL-1102 conservatively being something like a $200,000 a year annualized treatment cost, and obviously that's a very helpful price point for a new drug to be at. And there's no shortage of patients. Sad to say, there's about 300,000 patients worldwide with Duchenne's disease, and people have this disease for a long period of time, and so we think there is, sad to say, a very substantial clinical need for new therapies. So how competitive does this space look? If we're looking at putting a new drug in here, what does the competition look like? Well, by the standards of almost any pharmaceutical product, this is not hotly contested. A lot of the focus, as you can see in the top two rows of this table, has been on, for want of a better term, dystrophin restoration therapies, drugs looking to help patients make better and more dystrophin. And we've seen a few product approvals there, particularly from Sarepta, which has emerged as sort of the 800-pound gorilla in this part of the disease landscape. But down at the bottom of the slide here, in the anti-inflammatory and anti-fibrotic therapies, there's really not been an awful lot going on. And in fact, this slide is already a little out of date. FibroGen's drug there, pamrevlumab, recently had unsuccessful data in non-ambulant boys. So that potential competitor has removed itself from the field. They are still looking at ambulant kids, and so we hope they can bring some benefit here. But right now, the only real other company that's working in this non-ambulant space with a sort of anti-inflammatory therapy is this company Capricor. It's got a quirky cell therapy product they're developing. And as I say, what usually happens in diseases like this is people start looking at ways these therapies can be combined and co-administered. So the competitive dynamics here is really very, very different from most other industries. So we don't really see a lot of companies ahead of us in the game who stand to eat our lunch. We see here a field where ATL-1102 has a pretty clean shot on goal. And that's the opportunity we're looking to give the drug. In the interest of time, I'm just going to skip that next slide and just talk a little bit here about partnering, because of course, the simple fact of the matter is that it's unlikely that Antisense will be doing the heavy lifting of bringing ATL-1102 to market. What generally happens for companies like ours is that at some point we partner with one or more larger companies to bring the drug to patients. And that's certainly an effort that's very live. We're already doing a lot of outreach and a lot of discussion. My colleague Anthony Philippus is very busy out there pounding the pavement and putting the drug in front of people. And, this is a real kind of how long is a piece of string conversation, because those conversations can sometimes go on for quite some period of time. But in terms of what a deal might look like, we can certainly look to comparators here and say, well, what have similar kinds of transactions taken place at? And so the top part of this slide shows some recent licensing transactions that we think are good comparators for what we're doing. In particular, Capricor, a company I mentioned a moment ago, a year or two back, they licensed just the US rights for their product to a company called NS Pharma. And NS Pharma paid $30 million in an upfront payment, around about 700 million in milestones and undisclosed royalties on net sales. So pretty rich deal for just the US rights. And you can see here some other deals that we think look loosely comparable. And then Santhera there with vamorolone down the bottom. So these drugs trade for good money. And I think, look, every deal is different, every transaction is different, every drug is different. But I think our expectation here is that, a partnering transaction for 1102 should be, should be a high-value transaction. For comparison, we've also put some M&A transactions down the bottom, and it gives you a sense of what companies working in these sort of rare disease areas are typically valued at in an acquisition. And you see it's certainly in the billions of dollars rather than millions of dollars. So we do think there is enormous commercial value in what we're doing. I want to step back just for these last few slides to talk a little bit about the company, because we've been through some changes. And so I just want to briefly give you a picture of Antisense as it's now emerging from this sort of period of transformation. And then we'll open for questions. But this is some of the key members of the team as it stands. So the board along the top, Dr. Charmaine Gittelson, chair of the board, former CSL executive, 25 plus years of drug development experience here and in the United States. Dr. Gil Price, our other independent director, former Sarepta executive, company you've seen mentioned a few times in this presentation, and serial biotech entrepreneur, worked with a number of small companies. He's based in the United States and really just brings a huge wealth of experience to what we're doing. Of course, myself just recently joined as chief executive. And then on the lower part of the slide, you see some of the key senior members of the team. Dr. Anthony Philippis, Chief Operating Officer of the company. Philip Haynes, our Chief Financial Officer. Dr. George Takas, our Principal Scientist, very much the brains of the operation. George has forgotten more about antisense therapies and DMD than I think most of us will ever know. And they're not here today, but Dr. Andrew McKenzie recently joined us from CSL to drive our clinical program, and a really experienced drug developer. And behind these individuals, really one of the strongest management teams I've had the pleasure of working with in my career. So I think the company is tooled for success, it is equipped for success, and we're in good shape to take this forward. A word on financials. I'm afraid this is a little bit out of date already, because obviously our most recent financial statements are as at 30th of June 2023. At that point, we had just a whisker under $11 million in the bank. Now, since then, we've raised somewhere around about $11.5 million in gross proceeds from our recent financing round. Suffice to say, the company has some money in the bank now for what it's trying to do. We are well funded for the clinical trial and for the ongoing operations of the company. The bar chart here shows our quarterlies, our Appendix 4C filings with ASX. And as you can see, the cash burn's picked up a little bit in recent quarters, and that's entirely expected. That's all to do with starting up an international clinical trial. That's exactly what you'd expect. What I'd stress, though, is we spend most of our money on R&D. That's the red part of the bar. That's where we should be spending it, because that's what's going to add value for shareholders. And we run a pretty lean ship in terms of the G&A expense of the company. You can see here on the right, again, it's difficult to keep this updated in real time, but this is sort of generally as at 30th of June, and particularly our largest shareholder now, Platinum Asset Management, a sector specialist institutional investor, holds about 12-13% of the company as at the date of this slide. And we're incredibly grateful to have investors of that caliber supporting the company. And then finally, and this is really the slide I'd like to leave everybody, is this is an exciting time to be an Antisense shareholder. There's an awful lot going on. As you can see here from the first couple of rows on this slide, we've hit all the milestones we set for ourselves so far this year. We started our clinical trial in June. We've had some great data that we've been able to share recently in combination, preclinical data, but really exciting nonetheless. Coming up, we should pretty soon be able to talk to you about some of the preclinical work we've got going on in limb-girdle muscular dystrophy, aka dysferlinopathy. This is another form of muscular dystrophy that actually affects both men and women, and usually at a slightly later age. So really interested to see how this data looks. And then we have set ourselves the goal of finishing recruitment to our phase 2b clinical trial by the end of this calendar year, by the end of December. It's an ambitious goal, I have to say, but that's what we're shooting for. And as of today, we are on track. The study is moving pretty smoothly. So that's what we're shooting for. As we look into next year, we'll be working to try and get some of our existing data out there better. We'd like to publish our phase 2A study properly. We have an ongoing preclinical toxicology study, which people will be familiar with, and we should hopefully start to see some data from that in the first half of the year. And then, of course, the big ticket item, we will get that data from our ongoing phase 2B study, and that is really where the rubber hits the road for antisense, and that is going to be next year. So, ladies and gentlemen, as I say, it is a really exciting time for our company, and there is an awful lot happening. I've been absolutely thrilled to come on board. I've really been so grateful for the warm welcome from our team, from our shareholders, and I'm really delighted to be part of such an exciting business with so much potential in front of it. Thank you very much indeed. Now, we're going to open the floor for questions, and what I'll just briefly say, just for the recording, what I'll do is maybe just repeat everybody's question, just to make sure we get it on the tape. We've had a couple of questions emailed in from shareholders who couldn't make it today, who'll obviously kind of catch it on the recording. But let's throw it to the floor first of all and see if anybody's got any burning issues. So.
Yeah, I understand the company has been preparing for Europe.
Yes.
And I was just wondering how that was proceeding, given that it was not emphasised in the presentation.
Yeah, thank you. So the question was, what about Europe? I've been talking about FDA and about the US, and of course there's 200 and something countries in the world, and that's just one of them. And look, apologies, really, we think of ATL1102 absolutely as a global product. So we want to see this drug available to kids with DMD anywhere in the world. That's the goal that we want to work towards. For better or for worse, most people in the industry focus first of all on FDA. And for a couple of reasons. One is that it's one of the toughest regulatory agencies. If you can get your drug approved by FDA, there's a very good chance you can get it approved everywhere else. And secondly, it is the biggest commercial opportunity. It's not necessarily the most patients, but it's usually the highest price point. So for most drugs, the US market represents somewhere between 45 and 50% of the total revenue pool for that drug. So we all get a little bit hypnotized by FDA, and I must admit I'm guilty of that, but we are in parallel looking at our European regulatory strategy, and we're even starting to think a little bit, well, how are we going to get this drug up in places like Japan? The PMDA is the regulatory agency there. What are we going to be doing in China? How are we going to get this drug through SFDA there? So really, we want to position the drug globally. We do think of this as a global product, and perhaps I'd ask everybody to humor me. And when I say FDA, here FDA and other regulatory agencies worldwide, because we want to get this drug available to every kid who can benefit from it.
I can ask a few questions. Most of them might be covered in your report, which I haven't read properly, but what are the names of the staff that you've brought in from CSL? Sorry, what are the designations of the staff that you've brought in from CSL? So
they all work pretty much in clinical operations, that is, in the execution of our clinical trials. If you look back a year or so, the company had no active clinical trials. We now have an international phase 2B study running in 4 countries. And so a lot of the focus in the last few months has been building up that part of the business, building up the clinical operations function, run clinical trials, make sure we've got the right people to do that work. So that's really been where the growth is. Not all of them come out of CSL, but we've had 1 or 2 folks join us out of that organization. And of course, it's a great pedigree. You mentioned you had a couple of questions.
Oh, I can keep going, but throughout your presentation, there's obviously a few questions that popped into my mind. One of them's a bit wafty, but with regards to our CD target,
Yes.
which targets inflammation of DMD, are there other targets which also affect inflammation in DMD, and what percentage of inflammation would our CD49, whatever it's called, affect the inflammation from? So if we absolutely kill it in CD49D,
Yeah.
what percentage are we killing it in the inflammation of the DMD?
Yeah. Understood. So the question here was around, in a sense, how much of the activity of the disease are we knocking out by targeting CD49D? And look, there's a very, very, very long answer to that question, which will take us probably well into tomorrow, which I won't sort of test everybody's patience with. But look, the short answer is that the drug is selectively targeting one part of the inflammatory process here. So, and specifically, it is the mechanism by which white blood cells get into tissues. So clearly there are other things that happen as part of inflammation in this disease or in any other disease, but this is quite a good sort of choke point, if you like. If you're blocking the white blood cells getting in, you block a lot of the other things that go on. Now, we always have to be careful of these things. We don't want to block these processes 100%, because if you completely effectively shut off the function of the white blood cells, that can have very serious consequences for a patient. For example, their ability to fight infections. So in this kind of disease, and this is true of many other inflammatory diseases, multiple sclerosis, rheumatoid arthritis, psoriasis, you want to find the right balance, not too hot, not too cold. You want to suppress the immune activity enough to reduce the activity of the disease, but not so much that you expose the patient to a lot of things that are even worse. And so I think, certainly looking at the clinical data, looking at all the preclinical data we have here for 1102, it feels like we're in the right zone. We're not overdoing it, we're not underdoing it. And so that's what we're trying to achieve here, but it's a delicate balance.
I'll bang another one forward while you
Sure.
still got me. This was about the cohort of the 9 boys that we had, and you seem to suggest that when we go into the 25 and 50 ml doses going forward with 45 boys, that it'll be all randomised. We suspect that the 50 mils might pack a bigger punch, which may correlate to the weight or the level of inflammation going on in the boys. Is it completely random in that regard, or do we get a little bit of a...
So it is completely random.
A target.
So the question here was around just how we decide essentially who's getting what dose in the phase 2b study, who gets placebo, who gets 25, who gets 50, and whether there are things that might make a difference there in terms of how people respond to the drug. So first of all, this is completely randomized. That is to say, patients get allocated to the placebo, 25 milligrams or 50 milligrams, completely at random. And not only is it randomized, but it is also what we call double blind. That means we don't know at the beginning which patients. We only find out after the study is finished who got what dose. And we do that to remove any possibility of bias. This is something that regulatory agencies such as FDA or EMA in Europe or others really look to us to do in these kind of scenarios. They want to remove any chance for us to, for want of a better term, fiddle results. So it's a double-blind, randomized controlled trial. The significance of variables like weight or like inflammatory activity is not totally clear here. I think sometimes it matters a great deal in a drug. It's not completely clear how much it matters here, I would say. One of the reasons we're looking at 2 doses in this study is frankly because FDA has asked us to. And in general, they tend to want to see, in a study like this, more than one dose of drug administered. Partly to understand safety as much as anything else, but also to understand how efficacy varies. Whether we see much better efficacy with 50 than we saw with 25, I think just remains to be seen. I think we'll see in the data. The phase 2A study, all the patients had 25 milligrams. So at a minimum, the active groups in this study are getting that, and we'll see if we get a little bit more bang for buck at 50.
Yeah.
Just coming back to Stuart's earlier question about the molecule. My understanding is that the exon skipping products provide efficacy, or supposed to provide efficacy rather, based on replacing the dystrophin.
Absolutely.
How perfectly, I don't know, but let's say it does. So it heads the disease, it attacks the disease head on and saying, look, you will not be, your muscle fibres won't be damaged. Your cause, you've got the dystrophin there. And of course the steroids are anti-inflammatories, they're a bit broad spectrum, they're, other side effects, which is notable. For our product, for the ATL1102, am I right in saying that it doesn't stop muscle fibres from being damaged, because it reduces the severity of the damage because of its action on inflammation? And my question is, does the muscle fibres actually recover by administering ATL1102?
Yeah, no, I think that's a great question. So, again, the question, just to summarise, if I've got this right, is does the drug allow sort of recovery of function? Can it actually, in a sense, reverse some of the damage that's caused by DMD, or are we just in the business of slowing things down? Well, the most honest answer to that question is we'll find out with the clinical data to some extent. We'll see whether we get actual improvement in many of these measures, or whether we're just holding things still. I'll be honest and say it is often quite difficult to improve the course of these kind of inflammatory illnesses. If we look at, say, multiple sclerosis, we look at rheumatoid arthritis, which are, very, very imperfect measures, but they also have a strong inflammatory component. Not many drugs have been able to actually turn things around and restore function for patients. It will be great one day if we can do that, but that's still elusive for most diseases, like, where there is this sort of inflammatory component, where there is this sort of degenerative component. If we can hold things at bay, that would be a huge win. So, if we can just stop these kids getting worse, that alone would be incredible. And if we can, if we can slow the rate at which they deteriorate even, that would be a huge win too. So we don't necessarily have to, turn back the clock. We just have to try and stop things getting worse. And
So does that mean that greater emphasis should be placed on the combinational therapy?
Yeah, so question there again was around combination of ATL1102 with other therapies, and I think that's where the future lies in this disease. Right now we're looking at the drug by itself. We're looking at it as a monotherapy, as we say in the industry. And we have to do that because regulatory agencies like FDA are going to want to see convincing monotherapy data to approve the drug. However, the way we tend to treat these complex diseases like Duchenne's is usually with combinations of drugs, and sometimes that's tailored to the individual patient. But it's comparatively rare for patients in a disease like this just to be treated with one medicine for the whole course of the disease. I spent a lot of my career in oncology working with cancer drugs, and there we mix and match drugs in all kinds of ways. And I think because of the way ATL1102 works, because it has this quite general effect, and because, touch wood, it's also a fairly non-toxic drug, it's actually a very non-toxic drug, I think this has real potential to be what we call a backbone therapy. In other words, for patients to be on ATL1102 and then to dial in other therapies as needed. So I think that's where the standard of care will go if we're successful here, is we'll see this as a backbone therapy for kids with DMD, with other drugs kind of layered on top of it. Perhaps different drugs at different times in the course of the disease and different permutations. So, and I think that would be a great place to be. So, we've got time perhaps for one more question if anybody has any other things they'd like to talk about here.
So sorry.
Sure.
So in terms of financing, we have over $20 million in the bank. How far is the cash run rate as you see?
Yeah, we haven't put a date out there. I think we're just hesitating a little bit to put a firm date to our runway. What I would say is that we're covered for what we need to do over the course of the next year or so in terms of the phase 2b study. And perhaps I'll kind of just say that much today, that I think we've got the phase 2b study covered, but there's a little bit at the margins that depend a little bit on just how we prioritize some of the other things that are going on. So we haven't yet sort of put a date on our cash, but we're in good shape. We're in good shape. So, ladies and gentlemen, look, that brings us, I think, to the end of our first open house meeting. I just want to thank everybody again for making the time to come and meet here today. I'd also like to thank our hosts at Minter Ellison for accommodating us today. Minter's has been an incredible support to the company for several decades now, and just a tremendous source of wise counsel. So really grateful to them for putting us up this evening. And just say that we're going to be working on our website a little bit over the next few months. So I really want to stress that it's a great or it will be a great portal to go and look for information on the company. And also we have a company email address, info@antisense.com.au. So this is all part of us trying to really be as available as we can be for our shareholders. So some opportunities there to stay connected to the company in between these sort of interactions. Ladies and gentlemen, thank you once again for making time, and we'll look forward to keeping you updated as we go. Thank you.